Good morning, everyone, and welcome to the Phenotype Still Matters in the Genomic Era Symposium. Today, we're going to start the session with our keynote, Michael Tessler, who is doctor and assistant professor at the St. Francis College in Brooklyn, and did his Ph.D. and postdoc at the American Museum of Natural History. He is also has worked with systematics, phylogenomics, conservation, and environmental DNA. His topics also have include a stress response of jellyfish to robotic specimen samplers, ways to use leach blood meals to survey for rare animals, and the phylogenomics of early Metazoa. Recently, Dr. Tessler also co-authored a textbook named Phylogenomics, a Primer, second edition. Today's talk is titled Phylogenomic Node Instability, Node Support, and the Role of Morphology in Determining the Relationships Among Early Animal Lineages. So, Dr. Michael Tessler, thank you again for coming here and give us this talk, and please start sharing your screen when you're ready.
Is the screen good?
Yes, we can see.
Okay, great. All right, well, thanks all of you for coming. I'm super excited to be able to be here for this conference and to be able to get to share some of the things that I've been up to and the folks I've been working with have been up to as well. So my talk title, this kind of long, lengthy thing, gonna kind of break down what I really kind of care about with this. I was trying to give people some real information, but a lot of it is about kind of phylogenomics, right? The study of evolution and kind of large analyses using next generation sequence data, and where we're at with that, and some things that I personally think need work, and some things that might help, and some things that that we're kind of finding problems that need more work. So the first of these is morphology. So morphology, which is what this this group is particularly interested in, is the incorporation of morphology into phylogenomics. So it's generally ignored in phylogenomics, and so we don't necessarily know what its impact might be. Orthology, so coming up with how the kind of things that we compare, and kind of the genetic side, how we come up with our lists of comparable orthologous loci to check out. And so the way that this is done, it works pretty well in some instances, but often it's kind of questionable and hasn't really been worked out since kind of the first things that start to build on these happen. And then phylogenomic stability. So how good is the tree that you get in the end? Well, we really don't know. There's a lot of signs that have been pointing to the trees not necessarily being as great as they look. And so I'm going to at least get into a little bit on all three of these topics. So let's kind of jump in. Before I get going, there's a huge number of people that have helped with this work. And so I just wanted to thank them all before I get going, all the institutions that have been involved, the funding sources. And I particularly want to highlight Johannes Neumann, who is a Ph.D. student who's been critical in many of the aspects, particularly Johannes and Rob DeSalle on the sister to other Metazoa portion of this talk. So many of you, maybe all of you, don't know me. And so I just want to give you a little kind of clue as to where I'm coming from. My research is kind of all over the place. I love studying anything I can get my hands on if it seems interesting. So I've studied the conservation impacts of rock climbing on vegetation on boulders. I've looked at invasive carnivorous plants. I've looked at stress responses in jellyfish. But kind of over and over again, wherever I go, I basically always return to kind of phylogenetics. It's kind of the core of what I'm interested in, and it always is just fun and interesting to work on, whether that's the analytical side or whether that is the kind of more methodological side as well. So when doing phylogenetics, there are some basic steps that we all take. And the fact that you're in this conference and this symposium makes me suspect that most of you probably have some idea of where you're at with this. So we come up with a question, right? All science, we need to come up with a question. We need to do our background research, all that sort of stuff. We need to get our data. We need to align that data. We usually come up with a model if we're using molecular data. We then reconstruct our tree. We determine, the tree plus the support for the nodes and how stable they are, right? We want to figure out, is our end result good? And then, oftentimes we want, are actually interested in kind of the icing on the cake. What does this mean for, evolution of some trait, or can we do some additional analyses to get some more knowledge on that front? So what about phylogenomics, right? Phylogenomics and phylogenetics are pretty similar. The real thing is a scale level difference, right? So in phylogenetics, right, these terms are kind of fluid and overlap, obviously, but phylogenetics, typically when we talk about that, we're talking about, a few loci, a few genetic markers. Phylogenomics, we, hopefully are talking about hundreds, hopefully thousands of markers that we're using that are a lot harder to pick out, and often many more than thousands. Okay. So there are some similar things, but there are also some kind of big problems along the way with phylogenomics because phylogenetics, people have been chugging away at that for a long time. Phylogenomics is much newer that many people can be involved in this field and not just a select few labs. So there are some tricky things like getting data. So this is going to be a big thing that I focus on because what are the data that we're going to get? Obviously, phylogenomics implies that we're going to be talking about some next generation sequence data. So whether that's transcriptomes or genomes or whatever it might be, ultra conserved elements. There are many different things that often kind of fall under the broad strokes umbrella of phylogenomics, but big data sets, many loci, and particularly for this, focusing on kind of the genomic side of it. But what about other data sources? So in this case, particularly morphology, right? How does that play in here? This is kind of the big question. Does this matter? Should we be using it? Is it worth our time? Does it make any difference? Morphological characters are notoriously difficult to get, take a long time to get, and you only wind up with, dozens to hundreds typically of characters in a morphological matrix, as opposed to, tens of thousands potentially of markers that you can look at using genomics, and each one of those has, potentially many hundreds of characters within them. So a much larger kind of, deposit of information, right, of character information. But should we be combining those? We also need to determine orthologs. So again, are we doing an apples to apples comparison? Is, this thing we're calling a genetic marker, is that actually comparable between species 1, 2, and 3, right? That again is something that needs a lot of work. And then we also, and we'll kind of keep hitting on this, but we know that tree and node support / stability are not necessarily all they're cracked up to be. Most of our analyses in phylogenomics get wildly high support levels. So, 100% bootstrap support for every node. Okay, well, is every analysis equally good? No, it certainly is not. So how can it be that everyone is resolved as being, what that analysis indicates as being, kind of bulletproof or perfect? Well, it's kind of saturating at that level, which was less of an issue for, earlier phylogenetic studies, but is more of an issue for kind of these large-scale phylogenomic studies where we're not necessarily getting a huge amount of information out of our support metrics. So let's get on with it. So what's our question? So our question to start with is, what is the sister to all other metazoa? So it's a pretty cool topic, right? Metazoa. What is metazoa? Well, metazoa is just a fancy term for animals, basically. So animals in kind of, the broad zoological sense, right? And so this includes bilateria, basically all the stuff we think of as animals. So that could be a fruit fly, or it could be all of you sitting behind your computer desk. We have cnidaria. So these are jellyfish, anemones, corals, things that people are pretty familiar with. Then we get into things like comb jellies, which if you're not a zoologist, you may not know about. And so comb jellies look a whole lot kind of like jellyfish at a cursory glance, but they're really very much not jellyfish. They have a lot of unique features that quite clearly differentiate them. We also have placozoans. So these are very tiny. They're kind of amoeboid. They have very few of the features you would consider to be animal-like. They're just weird. And then we have porifera, the sponges. Yeah, sponges are indeed animals. And so these are the kind of major early kind of divergences in animal evolution. These are the major lineages that kind of make up those early splits. And so all of these are phyla, and bilateria is a larger group containing many of, again, the kind of main things we tend to think of as animals. So phylogenetic hypotheses for the sister to other metazoa. There are all sorts of phylogenetic hypotheses out there. There are, in fact, too many to show for this sister to other metazoa, which I'm going to kind of abbreviate as SOM in a lot of these slides. There are tons and tons of hypotheses. These are kind of the main ones, particularly these two on the left, which is either sponges or ctenophores being the sister to other metazoa. So who cares, right? Animals are cool, but does this really tell us anything? Yeah, it has major implications for evolution of a lot of different functions. So people have been looking this over, mulling this over, asking about it for quite a number of years, ever since ctenophores started to pop up as a likely candidate as the sister to other metazoa. And so this, if ctenophores are the sister to other metazoa, has major implications for the development of a lot of features. So was there an independent origin of the nervous system? Or were there two losses of the nervous system, one for sponges, one for placozoa, and then again for kind of the lineage that includes cnidaria and bilateria. So this is a major thing, right? Nervous systems are big. So are muscle cells. So are endoderm and ectoderm. So is digestion method. There are a number of things, including a lot of genes that have to do with the same sort of question. So this was pretty well summarized in a paper from 2019, and kind of looking at the two lineages that seem like the most important ones, porifera and ctenophores. What we find is porifera share a whole bunch of, so red being that they have something, and white being they don't, share a whole bunch of important characters with the other major kind of earlier divergence lineages in animals. They also share things with the kind of what is believed to be the sister group to Metazoa, which is the choanoflagellates. So as opposed to ctenophores, which don't share characters with choanoflagellates morphologically, at least not based on this study, and they share quite a bit with other groups like cnidarians and placozoans, as well as kind of this core group of characteristics. So just kind of walking through a few of these things. So how do these things stack up in the literature? And then we're going to get into a couple more things. So we looked through papers since the 90s, found a huge number of hypotheses again. And looking over these 3 decades, what we find is a shift largely from the most common hypothesis being porifera for many, many years, shifting to ctenophores and porifera more recently. And so this particularly starts with a kind of seminal paper by Dunn et al. In 2008, and is really the kind of mark of the shift from phylogenetics to phylogenomics, from a few loci to many, to hundreds or thousands of loci. So is this still up for debate? Well, going back, yeah, we basically have a 50-50 split on which hypothesis tends to be the most common. So it seems like these two, but people have put a huge amount of energy into this, really big data sets, always high support values in terms of things like bootstraps and posterior probabilities, very fancy analyses, add-ons to the kind of normal analyses, yet very little clarity. So there's a lot of room for figuring out more. So can we do better? Because, well, this was a pretty impressive list. Like, I'm not the most amazing human in the world, and some really big labs and important people have checked on this. But maybe we still can do better. Maybe we can add more to this question. And maybe morphology can help, because that's one thing that these studies have not really taken a dive into. So now we want to get our data. So again, typically these studies have used just nucleotide data, or they've used amino acid data, but ultimately the source being nucleotides. And so we want to incorporate morphology. Are they the same? Do they give the same results? What's going on there? Who knows? I don't know. You know, again, people have really not kind of brought these two worlds together all that much, which is too bad, because there's a lot to be learned by these broad strokes you can get from morphology, as opposed to nucleotides and amino acids that are much finer scale sort of resolution. So we got to make a whole bunch of trees, right? That's ultimately what we have to do. Dig into the literature, find a bunch of trees, and then make them combining our two data sets. So this is painful, it's slow, but it works pretty well. We get pretty good results. The methods have been pretty well worked out for tree building a long time ago, and just updated for larger and larger data sets. So we're going to focus again on this kind of case study in Metazoa. And this work is published particularly in this SysBio paper. Anybody's welcome to reach out to me if they're curious about it. And this has to do with combining phylogenomic data sets, so sequence data, with much smaller morphological data sets with typically moderate amounts of weighting, sometimes larger amounts of weighting. And about 10,000 total analyses were run, so a huge number of combinations between different data sets to see how robust the patterns we found are. And so just starting with molecules only, what we find is that basically all the data sets that we run wind up with CT, which in this case is ctenophores, as opposed to the original studies where most were ctenophores, but some were porifera. So in general, in studies that have differed between ctenophores and porifera, usually the phylogenomic ones that find porifera, often there are kind of add-ons to the traditional analyses that account for kind of issues in data sets, but ultimately wind up changing that particular node. When we look at morphology only, we find porifera is the most common sister to other Metazoa, with Placozoa being kind of, the other group that is also kind of, morphologically very distinct, and accordingly not surprising that it winds up coming out in some of these data sets. So what we did was combine these data sets together, and there's kind of a lot of information packed in on this slide. So don't focus too much on the details, but this is a kind of combination of morphological data sets plus the molecular data sets. And we tried exemplar data sets using just 6 taxa and 11 taxa, so we could kind of control the taxa specifically that were used, as well as the full original data sets. And we did this in likelihood as well as in parsimony. And so the flip, again, is going from the molecules, which is ctenophores, typically to porifera for the flip that occurs. And what we find is even a few hundred morphological characters. If we look, these were hundreds of thousands of characters. These were, dozens to a few hundred characters. We find that incorporating them with often modest amounts of upweighting leads to a flip at this node to porifera rather than ctenophores. And so this scale is basically 1x is not weighted, 2x, 10x, 10 to 100, and greater than 100. So this is pretty heavy weighting. So if you see this color, it means the data set was pretty stable. If you see this weighting, the data set was really not stable and flipped to porifera very easily. And what you see is basically most of the data sets, aside from a couple, could be flipped with very, pretty modest amounts of data. We also find that having fewer taxa or parsimony has less stable results, which makes sense. We know you get more issues with long branches and things like that that tend to be broken up by having more taxa. So that helps provide a level of robustness to these analyses. Parsimony is not surprising that it's easier to flip. Parsimony has a tendency to kind of emphasize uncertainty rather than model-based methods. And so it's not surprising that it takes less to change its answers around. So well-supported nodes in phylogenomics are not necessarily stable. Again, all those studies with ctenophores had very high support, usually full support. So it doesn't take a lot to shift those. So there's a bit of a kind of David and Goliath story where a really small morphological matrix can impact really large phylogenomic data sets, indicating this is still something we should be considering. These nodes aren't stable, so even a few characters are still really important despite these huge data sets. So we've been kind of continuing this work, and we're curious about things like, where do ctenophores go when you add this morphology in kind of larger and larger data sets? So we've been using a genomic data set that we've kind of compiled ourselves with over 10,000 loci, and we've just started trying a couple different weighting schemes. So here we have 50 and 100x weighting schemes. And what we find is without morphology, you wind up with ctenophores as the sister group, okay? So not shocking. That's what we kept finding. When we add 50x morphology, we also have a fairly modest shift, basically a flip between periphera and ctenophores to periphera being the sister to other Metazoa. When we do 100x, which in my opinion is a little bit heavy-handed, it's very subjective, but it's still worth exploring, we find a very different answer. Ctenophores wind up being sister to bilateria, which is not a hypothesis I'm trying to support or endorse in any specific way. More the point is, this is very unstable where ctenophores go. They can be flipped around and moved to multiple parts of the tree without necessarily a huge amount of kind of difficulty to make that happen. So still not stable, right? Even ctenophores, not stable. Everything else is much more stable in our analyses. Ctenophores jump around. So again, evolutionary implications. Ctenophores hop around. We don't know what's going on. What we do seem to get is that in phylogenomics, adding morphology suggests ctenophores are not the SOM and that Placozoa probably is. Sorry, not Placozoa, Peripheral, too many words with P's. Peripheral, the sponges. We also find Placozoa is, a more recently diverged group, kind of like it's being shown here, which means it's probably a severe reduction in its various features. And so this has major implications, again, as suggesting that no, the nervous system didn't evolve more than once or wasn't lost in two separate lineages, because we would find ctenophores are flipped with periphery in general. And accordingly, you have kind of a single gain for these major features like the nervous system. So where to go next? Well, other lineages should really be explored for this. These are small data sets. Our kind of best data set is something which was used in that most recent analysis on where ctenophores go. It's 75 characters. It's tiny, right? Whereas you can get a lot more characters for certain things like arthropods if you work on them. So those would be a great next step, groups with a lot of kind of discrete features. Genomic morphology is a really kind of interesting area that needs further exploration. And then we also need to be combining our work with kind of other strategies. So we know that cat models combined with reduced amino acid alphabets have made flips to things like the sister to other Metazoa node. So what happens if you combine those with morphology? What happens if you combine gene presence absence, gene content, or indel coding, the presence of gaps, if you code those? There's a lot of information, a lot more characters that can kind of be mined out that might provide some level of robustness and support that we haven't yet found clearly. So the SOM is still not all that clear. So what can we do? Again, we just kind of went through morphology. But we probably need to start exploring phylogenomics in other ways because, morphology, people have been studying that for, in the SOM question specifically, not in a lot of other groups that are poorly understood for morphology, but with the SOM, people have been looking at characters in these things for, a few hundred years. So it's going to take a lot more work to even get a few more characters there. So we need to kind of eke out the most, get the most out of every character we already have before we kind of, move forward. So there needs to be a lot more done on the kind of phylogenomic era. So the first thing is, again, can we trust support? And all signs are pointing to, no, you can't trust support. Because again, the SOM kept being highly supported in lots of other people's analyses, and we can flip it with a couple hundred morphological characters. That doesn't seem like that support has a huge amount of meaning to it. So we want to jump in and see, node support and stability. Can we trust these findings? And so people have looked at tree metrics like the retention index to assess the quality of a tree. People have used resampling techniques like bootstrapping and jackknifing, particularly bootstrapping, as we've been talking about, and posterior probabilities as ways to assess node level support. So why might well-supported nodes be unstable? Well, bootstraps might not be meaningful, right? Our kind of main metric that most people use, bootstraps or posterior probabilities, they might not actually be meaningful, particularly in phylogenomics. They tend to saturate very easily with just a little bit of evidence kind of pointing in one direction that they wind up looking very, very stable. And we know this is true for our kind of sister to other Metazoa case. So what we wanted to do is check out some simulated and real phylogenomic data sets and see, are higher numbers, high bootstraps, are they actually meaningful? Are higher numbers better, right? And so almost surely, higher numbers probably are better, but really are high numbers meaningful might be a better way to put it. So we simulated 100 loci using a mixed ratio of kind of fixed topologies, so always the same relationships, versus random topologies, always different relationships. And in all of these, we varied branch lengths and we simulated amino acid data from these topologies. We then concatenated them and ran trees with bootstraps, and we varied the level of missing data. And so what we find, so this is level of missing data, no missing data, 25%, 50%, 75% missing data. We find that no missing data is more stable, which isn't surprising. But again, if we look at the percent that is fixed data, we have 0% fixed versus 100%. So 100% means all the topologies that the amino acid data were simulated from should be identical or are identical, which means this should be at 100, right? But something where it's 0 means all the topologies were different and random. So combining them, you would expect a low score. And so it looks like you do get kind of a lower score. But when you kind of zoom out, it's actually really, really bad. When you actually make the Y scale bar from 0 to 100, you find all of these, regardless of missing data, are very close, at 90 to 100 across their whole range, even when you're using totally random data. This indicates this is not good. Don't use it. Or be highly skeptical, maybe, whatever it might be for you. But the simulation is very, alarming. So we wanted to try this with something similar with, empirical data sets. And so what we did was we took genomic data sets and we scrambled the taxon labels for them. So all real data, but mixed up in terms of, what actually corresponds to a given species that was used in the tree. And what we find is you get a lot worse support scores, but you do still find some very high support scores even for these data sets. So it's not as worrisome, but it's still worrisome, right? You might have trusted one of these nodes, even though they're just random. So kind of how problematic this is depends on your personal opinion on the matter, but it's at least something that you should be starting to be skeptical about. The simulations are very scary. The kind of empirical scrambling is less scary, but still worrisome. And we know from real data sets like the SOM, 100% does not indicate genuine stability. So there are some solutions like some concatenation analyses, sensitivity analyses, and we're working on some other analyses for building tree level support, and testing those out that hopefully we'll be able to finish up in the not too distant future. So I'm going to finish up going through the kind of last component, which is the molecular data. So our molecular data, again, we have so we have a lot of data, right? We have hundreds of thousands of characters to millions of characters in kind of our more recent analyses that I showed you with the movement of the ctenophores. So can we get more out of them? Are these actually as good as they can be? And the answer is probably not. So orthology is probably not as well set up as you would assume it is, given how much it's used over and over again in these types of works. So orthology in general, it seems very straightforward. Probably many of you in a class at some point have seen a diagram like this explaining orthology. And it's like, oh, this is simple, I get it, whether it's an ortholog or a paralog. In reality, orthologs wind up being really confusing to talk about and not being so obvious. There are, analyses are really complicated, and accordingly, or evolution is really complicated. There are a lot of divergence events. There are a lot of kind of, gene death events. And accordingly, it winds up being very, very complicated. So most people have kind of skirted this issue by just using single copy loci. And I put single copy in quotes because this tends to be on the scale you're looking at. Most things have had kind of, duplication events at some point if you look broadly enough across the tree of life. So most loci are actually multi-copy loci, and they are really complicated like this. So these are some leech antistasins that I worked on recently with a student, Raphael Iwama, and they're complicated. They're just very, very complicated. And so we wanted to check these out and see, is there a better way to deal with these things? And so we came up with 3 genomic level data sets. And what we find is multi-copy data sets, multi-copy loci far outnumber single copy loci. So in some of the data sets, there is about a 1-to-1 ratio, but in many of the other data sets, it's a much, much higher ratio. It's about, in these almost 10 times more multi-copy loci compared to single copy loci. So we came up with kind of a complicated way to test what is the best way to pick between multiple copies to ultimately make a kind of single uniform locus for incorporating those multi-copy loci. So a lot of people have done things like this, but they tend to pick one metric for picking between the multiple copies, and it doesn't necessarily, it's not based on anything. It might be a good idea, it might be a bad idea, but it's never been tested. So we tested a few dozen different metrics for the speed, for the tree scores they built for their individual gene trees, and for the total evidence analysis, and as well as some other metrics that we're exploring that I mentioned before, like for these tree score types of things. So what worked best? So in this case, red is good, yellow is bad. So here, runtime, you want it to be as fast as possible because these analyses take forever. And and red is good. So here, very little would be red, whereas for something like tree score, the larger it is, the better it is, right? You want a high tree score, at least for something like likelihood, right? You want the opposite in parsimony. So a little bit confusing, but the main point is between all the metrics that we ran, what we find is that you have things like maximum likelihood tree score. It's an excellent performer in terms of quality, at least in the way we were judging things, in large part because we were interested in things like tree score, but it takes a long time. And we were using a shortcut method, FastTree, rather than something like RaxML or IQTree to do the runtime. So this was much, much higher if you wind up using one of the more genuine tree building algorithms for each of these gene tree combinations. And you have to test between many different combinations, so you really want to focus on speed too. And what we find is things that are kind of just changes in alignment length and things like that can often be a much better predictor and take almost no time, or sorry, as good of a predictor pretty much, and they take almost no time. So they are a really good way for picking between paralogs. And so again, why is this important and why does this relate to the SOM? Well, basically it relates because the SOM shows that even a few characters, the sister or other Metazoa, even a few morphological characters can change things. So if we can change the orthologs that we are using, we have thousands of different orthologs. So if we're changing even 5% of them, we're making really large changes compared to what we're able to do in terms of gathering new morphological data. So it's really important to kind of squeeze out as much from morphology, squeeze out as much from molecular, and ultimately get at the best result you can. And so when we made our concatenated analyses using the kind of best metrics we had available, we find that these are generally relatively robust data sets. But we find the things in purple, the level of support shifted, and we also find that the nodes that are labeled in black, there is actually a shift in the positioning occurring there. So even small things matter, particularly for the nodes that we tend to care about because they tend to be poorly resolved, things that people go back and forth about, like the SOM node. And accordingly, anything you can do, if you can improve that, would be a major, major advantage. So we can improve things by adding morphology, hopefully by coming up with better support metrics or using better support metrics in the future, and by improving our way of doing orthology, which at least hopefully, when these things are combined in the future, should be able to help with questions like the SOM and other important nodes that we don't have a lot of confidence in, to help to try to get at ultimately more confidence-inspiring solutions. So to kind of go through and summarize what we just kind of went through in general, phylogenomic trees can be highly unstable, even if they're really, really well supported. So just because you see 100 on a node doesn't mean you should say, oh, this is what happened. This is how that group evolved. I mean, I wish we could, and hopefully in the future we'll get closer to that point. But we know at this point, a lot of these solutions are not stable. And this is kind of highlighted for the sister to other Metazoa. And what we find is adding morphology, even a small number of characters, can be really important to resolving this. And what we find is that we get support using morphology for porifera sponges as being the sister to other Metazoa, which has important implications for things like the nervous system, not having evolved multiple times. And so it winds up being a very, it's much more in line with morphology, which isn't surprising as that's where the data set comes from. So I wouldn't say we're done with this question at all, but I do think morphology is helping to add into the piece of the puzzle that really wasn't there and really is an important data source, given that we know a lot about morphology and have spent a long time learning about it and coming up with characters that are really important to animal evolution, plant evolution, microbial evolution, whatever it might be. So orthologs, we can be doing these better. We should be doing these better. We have a little ways to go. Hopefully some of the methods I'm working on will help put that more in the right direction. And again, bootstraps in phylogenomics are something you need to be highly cautious about. We need better methods for addressing stability. And many other people are already starting to talk about this, and I'm sure there will be better methods in the future. Again, there are some good methods like radical that can help bypass some of these. So we've come a long way in phylogenomics. Adding morphology helps, but we still really have a long ride ahead to get at some of the really important questions in evolution. So you all still have lots of work to do, which is good. It keeps us employed. So with that, again, this paper by Neumann et al., 2021 in SysBio, you should check that out. That goes through a lot more detail about the sister to other Metazoa question, and hopefully we'll have more things in the future. So with that, thank you all so much for inviting me to be here, for coming to my talk. It's been a pleasure, and I'm happy to answer any questions.
I am clapping because the audience can. Sophia, help me out.
Thank you so much, Mr. Tessler, for this talk. It was amazing. I know everyone is thinking the same. And we have already some chats going on. So Acacia says, excellent lecture, Dr. Tessler. What do you think about the use of ontogenic data in an attempt to reduce homoplasy in morphological analysis? Does it make sense? Sorry.
Yeah, the ontogeny is always when questions get serious. Yeah, I think that anything you can explore in that direction, that's obviously something that is really interesting, provides an additional level of information that you don't necessarily find from everything. For kind of the question that I was working on for the sister other Metazoa, a lot of things, they're just really, really evolutionarily divergent, right? A lot of these lineages split apart hundreds of millions of years ago, or they did split apart hundreds of millions of years ago, which just makes it very hard to come up with character sets that are larger. But I 100% encourage people to check out ontogenetic data or build their own data sets using ontogeny. Yeah, I worked with when I was doing my PhD, there was a postdoc in the lab who was very interested, Joe Wolf, who was very interested in that question. And I think that there's still a huge amount to explore in that direction. And yeah, it's a great question.
While we are waiting for our attendees to type more questions and answers, I have one of myself. I am very interested in support metrics, and I'm actually preparing manuscripts on that topic, and therefore the question is based on my current research. As bootstrap values are not actually direct measures of support in the sense that they do not correlate with the optimality criterion, like, for example, likelihood ratios do or Goodman-Bremer indexes do, it's known that bootstrap values do not directly correlate with these notions of support. This question is open to debate, and the reviewers of my paper certainly agree with that point of view. This is open to debate. However, what is not open to debate is that bootstrap is not designed to do anything else than to calculate the frequencies of nodes using different evidence, not the same set of evidence as the original analysis, but a different set of evidence. In that sense, would it be worthy to thinking about the graphics that you showed us about that very preoccupying situation of how we cannot predict stability of nodes based on bootstrap values? Do you think, or maybe you maybe already have that would be worthy to analyze how that would behave in comparison to, for example, the likelihood ratio test or the Goodman-Bremer index instead?
Yeah, 100%. It's a great point. And when you talk to people who actually know this stuff well, as you clearly do, there's a lot of complexity and a lot of argument over what we should be doing. What I can tell you is that what people are doing is bootstraps, right? Or posterior probabilities. That is 99% of the way that people currently assess trees. So basically everything that has ever been, given the label of support has a problem, right? Which is true for all these analyses, right? We don't know the answer on phylogenetics, which I think is what makes it fun, right? Nobody was there, and even if you were, how could you pay attention to all these organisms across the globe all at once? So we don't know, right? There's a lot we don't know, and a lot you have to kind of play with the data and see where they take you. So I think this was kind of a is a first pass. This is what people are doing for the bulk of things. This does not mean this is the solution that should be used, and whether that's any sort of metric around likelihood or Bremer or whatever it might be, right? Or the concatenation analyses that I mentioned, there are better things out there, right? There are. I would personally trust something from Radical, which is not commonly used, more than I would trust Bootstrap by a long shot. That doesn't mean people are using it, even though it's probably better, right? And so the same could be true for many different metrics, and we're messing around with things with Bremer support and things like that, and certainly other things would be great. And you are more than welcome to, I think you already have my email, to reach out. Anybody is to reach out if you have other questions or interests in some of these things. I think a lot like the support, it's a big question, and it's a something that deserves so much more attention and working with more people is always better for for trying to solve these problems, right? But yeah.
I appreciate that answer. And I'm not going to be monopolizing the talk, although I could potentially be talking to you for hours and hours, but thank you very much for that.
Well, you got my email. I'll be there.
I will bug you. Sophia, can you read the next question, please?
Yes. Well, the next one is not a question. Is from Maite Soto. She says, I'm a teacher assistant in my university's taxonomy class, and I'll make sure to share this with my students. You raised very interesting and very important points. Thank you.
Oh,
and
that's
the
so
next
nice.
question is...
well, first, thanks to Maite. I appreciate the nice comment. It's really nice.
And the next question is from Guillermo Delia. He says, is there any evaluation of regulatory bias aimed to assess the homologies of the morphological characters used? He means the contending main lineages are quite different in morphology.
So did we analyze that specifically? No. Are these data sets contentious? Yes. Which I think is the kind of main point of that. I mean, like I said, for orthology with molecules, there's a lot of debate for that, right? Which is very bioinformatically kind of informed. The same is true for morphology. People have been debating about that forever with a lot of these characters. And I think a really important kind of question around that's been back and forth, just batted around the courts, is with things like the choanoflagellates and the choanocytes in sponges. Are these the same things? Are these not the same things? And people argue strongly in both directions and come up with good points. But so there's always a level of uncertainty, and homology at whatever level, whether that's orthology or something else, is always kind of the crux of what we do, and always something that we will be fighting about until, for all of our careers, because it's not something that's easy to know. And so we do our best. And I think these are pretty good morphological data sets. And we've kind of, like I said, we've been refining them even more, and we're kind of down to a streamlined 75 character matrix that I think works pretty well and deals with this stuff pretty well. But there certainly is a lot of room for interpretation along a lot of these lines. And I do think there are things you could do with Evo, Devo, or whatever it might be that might help with that.
Thank you very much for your answer. I hope Guillermo really understood what you said and solved his question. Next question is from Mu'min Michael. I'm sorry if I pronounce it wrong. Can shape data from geometric morphometics be used as morphological data sets in combination with gene data sets?
Yeah, and there are a lot of people who work on that. So certainly that's an active kind of, promising area of research over the last, I don't know, handful of years to decades, something like that, where people have really started to play with that and try to incorporate it. It's not something I have specific expertise in, and unfortunately it wouldn't, as far as my understanding, work well for the SOM question. But it would work well for something like I'm working on mammal skulls or, arthropods or whatever it might be, something that is a lot easier to measure and, has more identifiable points of homology. So yes, you can. I'm pretty sure that's well integrated into TNT. Somebody else might know better than I do on that front. But yes, I'm pretty sure you can, although I think the methods are still very much actively evolving. And that would be a great way to move forward with some groups.
Okay, thank you very much. There is no more questions in the chat right now. So if someone wants to go to the next talk, that is by Stefan Richter, so you can move to the next link. And if you want to ask any more questions, either in Spanish or English, you can put it in the chat, and we can stay here a little bit longer answering those questions.
Yes, and I to help Sophia, I put the link to the next talk in the chat. And I would like to remind you that it starts at 9:50 sharp, even if we are here. So let's give a few moments to see if somebody else has any other questions, but probably people just want to move to the next talk so they don't miss it. Meanwhile, Dr. Tessler, amazing talk. We are through difficult times. I know that it's hard on all of our speakers to find enough time to put a talk together, think about it, come here, endure our numerous emails, and give your presentation. Thank you very much for your time. We could not appreciate more your presence here. Thank all the attendees for coming. And if we don't have any more questions, I would like to ask Natasha, that's helping us from UNC Charlotte Sand, to close the session. Bye-bye, everyone.
Thank you.
Bye, everybody.
Bye.
Thanks so much for having me.